everyone. I hope you're having a good week. I wanted to go over with you what the expectations are for the unknown project. And the unknown project that you guys will be doing this semester is going to be a little bit different than what we usually do. And that's because obviously we're not in class, so you can't do this hands-on. So we're going to have to do this as a remote project. And so the purpose of the unknown project is to identify unknown bacteria. And if you think about why that's important, right? If you have a patient and they come into the hospital with some type of unknown infection, it's important to know what type of infection is causing the disease so that you can prescribe the proper medication. This is also important in industry and in research, right, to help identify unknown bacteria. Let's say we wanted to study different bacteria in the soil. Well, we would first need to identify such bacteria first. And so in this experiment, you would have a mixed culture of two unknown bacteria. And so the way that this would work would be that in normal class, we would give you a broth, and your broth would contain a gram-negative and a gram-positive bacteria. And your goal would be to determine the identity of your gram-positive and your gram-negative. And so the first part of this experiment would be to isolate two bacteria, right? You would need to isolate the bacteria and separate them from one another because when they're in a broth, right, when they're in a broth, they're going to be mixed. So before you can even begin to study those bacteria, you would need to isolate them. And if you think back to the first technique that we do for isolation, you might recall that to isolate colonies of bacteria from a mixed culture, that our technique that we use is a streak plate. And so the first part of this experiment would be looking at a streak plate. Now again, had we been in class, you guys would be given the broth and you would be doing the streak plate hands-on. You would be actually streaking for isolation and we would be testing your technique. Now, however, obviously you're not in class. So for this project, instead of you setting up and getting the isolation yourself, the pictures for the plates are going to be provided for you. So to get your pictures, I'm going to walk you through where you find those. And I'll walk you through on Canvas in just a minute. So I gave you instructions here and it says under modules on Canvas, go to the unknown project section and click on unknown project info. And this will give you a list and you'll find out which organism you are assigned. Once you find out your organism number, then you're going to find the corresponding picture. So let me show you what I mean by that. So here is your Canvas page. I have it in student view so that you can see what it would look like for you. And so you're just going to click on modules and you're going to scroll down to the module for the unknown project. So it's going to be all the way at the bottom. And so here is your unknown project module. So there are several parts that have the directions. So these are all the different directions. And then here are the places for your submissions. So if I go to unknown project info and I click there, you'll see this page and notice it says click here to see the list of which unknown number you are. Then use that number to get the appropriate nutrient agar streak plate and the appropriate McConkey streak plate. So I would click here and it's going to pull up a list. So here comes my list. And so you would find your name. They're alphabetical by last name. And let's say, for example, that I was assigned number 1. So let's say this was me and I was assigned number 1. So you need to find your name on the list and make sure that you are using your number. Now, some of them are 1 through 32. Other people have 101, 102, etc. Make sure you're using the right number. So let me scroll back up. So let's say I was number 1. So I'm going to go back and I need to find my number 1. So there's 2 places I need to get pictures from. Here's one. And so it's going to bring up a Google Drive. And notice that this is the nutrient agar plate. And if I'm number 1, I need to use this picture. Make sure you use your number. So this would be my picture. I would download it and then use that for my report. I would repeat the same process and do that for my McConkey auger as well. So I would click here. Again, it's going to bring me to a Google Drive. And then here is my McConkey. Notice number 1, this is my McConkey plate. So I would download that and I would use that plate for my McConkey auger. And so this would be where you would get your pictures for your streak plate. So instead of doing your streak plate yourself, you're going to use these pictures, and those streak plates were done for you, and you're going to describe your 2 streak plates, both your nutrient auger streak plate and your McConkey auger. And so notice it says download those pictures and then use those for the streaking part of this technique. Now, you're going to describe, there will be 2 colony types on your nutrient auger. You need to describe the 2 types of colonies. Remember that your nutrient auger is going to allow both bacteria to grow, gram-positive and gram-negative. And so you should have 2 different colony types on that plate, and you're going to describe those 2 colony types. You can call them colony A and colony B. Then you're going to describe your colonies on your McConkey auger. What color are those colonies? What does that tell you about that bacteria? So that is where you're going to find the first part of your experiment. So for your McConkey auger, make sure to go back and review. There was a section in the Lab Practical 2 question set that discussed the McConkey auger. The McConkey auger was not on exam 2, but it is going to be, it was included in that because you needed to do some background research on what McConkey is used for. Now, when you go to describe your colonies for your nutrient auger plate, for example, notice if I scroll down to the report section, it talks about when you describe your colonies, it says look at figure 2-3. So this was related to your environmental cultures. And so go back and look at that image. They describe the shapes of the colonies. So are they convex? Are they umbani? Do they have smooth margins? Are they lobate, etc.? And so you want to use that figure to help you to describe your 2 types of colonies. And so you want to describe thoroughly what those colonies look like. So when I'm recording what those colonies look like, I need to fill out an identification test sheet. And so let me show you where that is on Canvas. So under modules on Canvas, remember that I just showed you, here's the unknown project info. That is where you find what number you have and you get your 2 streak plate pictures. This is the directions. This is what I've been covering in the video, that Word document that I've been showing you. And then you'll notice some additional figures. So we have this identification test sheet blank, and we have this identification test sheet example, and then an unknown identification flowchart. We will come back to the flowchart in a minute, but first we're going to start with our identification test sheet. And so you click on that and it's going to bring open a page. So here is an example of the blank test sheet. So for each of the tests in this project, you're going to need one of these identification sheets. So when you start out, you're going to have one for your nutrient auger streak plate. You're going to have one for your McConkey auger streak plate. So name of the test, I would type in nutrient auger streak plate. Purpose of the test. So again, remember back when I talked about every experiment, I always had the purpose of the test. So you want to go back and review the purpose. Why do we do this test? What is this doing? The procedure that's actually followed. So in this part is where you're going to describe your technique. Now, you don't have to get very specific in terms of all of the steps of aseptic technique. Assume that the person that is doing this knows general aseptic technique. And what I mean by that is you don't need to mention that you're going to vortex and then you're going to take your cap with your pinky and ring finger, flame the opening of the tube, flame your loop, dip it in the, wait till it cools, dip it in the broth. You don't have to go through that much detail. That will make more sense when I go through an example with you. So just be aware that it's not so detailed that that you're just telling me every step in aseptic technique, but it also needs to be detailed enough that it tells somebody how to do that. And what I mean by that is it should always include, are you using your loop or your needle to inoculate whatever tube it is or plate? How long are you incubating? So how long do we typically incubate that test? Things like what temperature do we incubate this test? Some of our tests go 48 hours. Some of our tests could be done in as little as, remember, 2 hours if they're a rapid test. So you want to include at least that much detail. So what type of tool do you use for inoculation? What temperature? How long do we incubate? All of those things need to be in your procedure. And again, this is going to make more sense when I go through an example. Make sure that your procedure is in your own words. You should not, again, you should not copy and paste from some source on the internet. Use your own words. Go back and review your notes and use your notes to help guide you for what you should put in your procedure. So that's your procedure. Your test results, so test results including the appearance, so color drawings or digital photos, and any unexpected observations. For this, I'm going to allow you, so remember I told you that you're going to download your nutrient agar streak plate and you're going to download your McConkey agar. All you need to do is either insert that picture into this document or you can draw it. But if you do decide to draw it, make sure that it's colored and that the drawing is detailed enough that I can see what you're describing. So you can either draw it or you can insert that picture there directly. Now you might need to resize it because you might go to insert this streak plate and it might end up being huge. The file's large so that you could see the details. Just resize it down to size. If you need to, you can use this as a PDF. It doesn't matter how you decide to do it as long as you get a picture in this part of the identification sheet. And then the last part is the interpretation of your test results, including a full description of what products, if any, were detected and how those products contributed to the final appearance of the test. Indicate any reagents or pH indicators that are used. Also indicate substrate, enzymes, and products. So that part, that detail, that interpretation is more for your biochemical tests and applies less to your streak plates. So what you want to put in your interpretation for your streak plates is, let's say I have my nutrient agar plate. In my interpretation, it's going to look a little bit different. I would include things like, did both my gram-positive and negative grow on this plate? And then this would be where you would describe those colonies. The test results, you have the picture. In the interpretation is where you're going to describe those colonies. So colony A is convex, round, smooth entire edges, etc. So that's where you're going to describe what that looks like. For the McConkey plate, for your interpretation, you want to tell me, what do the different colors mean? What type of bacteria will grow on a McConkey plate? And then what do those colors tell you? So again, review your McConkey agar plates for your interpretation. But all the other tests in terms of your interpretation are a lot more straightforward because they're simply biochemical sheets. So you will need one of these for your nutrient agar plate, and you will need one of these for your McConkey agar plate. Now, let me go through an example. Again, this is going to look a little bit different than what your ones for your plate would look like, but again, it still gives you the level of detail. So let's say that I'm doing an oxidation fermentation test, an OF test. So I need to write the purpose. The purpose of this test is to determine whether bacteria oxidize and or ferment glucose. Again, those are on the slides. I gave you those all throughout the semester. The procedure. So this is the amount of detail. So obtain 2 OF broths and a dropper with mineral oil. Using aseptic technique, Stab inoculate 2 OF broths with a needle. So again, remember that I want to be specific with which tool I'm using, and I specified using aseptic technique so that I didn't have to describe every little step. Making sure to go almost to the bottom, about 1 centimeter from the bottom. So again, you want somebody who's looking at this to know how you did it, right? And so you want to tell them that they don't go all the way to the bottom. They need to go about 1 centimeter for the bottom. For one of the tubes, layer enough mineral oil over the top to seal out the oxygen, about 1 centimeter worth. This tube is your anaerobic tube, and the one without mineral oil is the aerobic tube. Make sure the cap is loose and place both tubes in the 37-degree incubator. Incubate at 37 degrees for 48 hours. The other thing that I really want you to get in the habit of, and it's listed in the instructions, is writing for scientific writing. And what I mean by that is you shouldn't say I, first person, third person, we, us, etc. You don't want to say I did this. Notice that it's very blunt. So obtain 2 OF broths, stab inoculate. So I'm not saying I at all. It's not in first person. So it's going to take a little bit of practice for you to get used to writing this way. But it's a good skill to learn because that's how scientific writing is written. It's not written in first person. So the test results. So here is the test result. Okay, so this is my picture of my test. There were no unexpected results. The aerobic tube without mineral oil was yellow on the top and green on the bottom. The anaerobic tube with the mineral oil was green throughout. So I'm not interpreting it yet. I'm not saying what the yellow and the green means. I'm just simply describing the result. What does it look like? Now here's your interpretation. Again, this is your biochemical sheet. So all of your biochemical sheets that you've used so far are going to help you with this section. Again, it's not written in first person. It's written in scientific writing. So it says, for the aerobic tube, the tube was yellow on the top and green on the bottom. This means that the organism can oxidize glucose, and notice I put in parentheses the substrate, using various endoenzymes, those are the enzymes, to produce the products, which are acids. The acids produced by this reaction lowered the pH, causing the bromothiamyl blue, the pH indicator. So again, I'm listing my pH indicator. Some cases it's a pH indicator, some cases it's a reagent, to turn yellow. So I'm talking about why the color changed. The bottom of the tube stayed green with a neutral pH, which means that the organism only used the sugar aerobically. For the anaerobic tube, the tube was green throughout. This means that the bacteria could not ferment the sugar to produce acids. The pH remained neutral and the bromothiamyl blue stayed green. Collectively, so I'm summarizing, collectively these results indicate a positive result for the oxidation of glucose, but a negative result for the fermentation of glucose. This organism is a strict oxidizer of glucose. So that's the level of detail. I'll go through and I'll discuss these more in just a little bit, but this is just to walk you through to give you an idea of what is expected on these identification sheets. Again, this one is going to look a little bit different than your streak plates because your streak plates, obviously we don't have biochemical sheets for. So it's more for the interpretation. You're going to interpret what's on your plate. So do you have isolation, and then describe what the colonies look like and why they look like that in the case of the McConkey agar. And so that's your identification sheet. So when you finish that for your experiment. So once you've made your identification sheets for your nutrient agar and your McConkey, you're going to have 2. So one for McConkey, one for nutrient agar. You need to submit those onto Canvas. And so let me show you where that is. So if I'm on the modules page and I'm on my unknown project, notice that there is a page here that has unknown project submissions. And so the first one says nutrient agar and McConkey agar streak plate ID sheets. And it tells you the due date. That's the date that has to be submitted by. Of course, you can always submit it earlier, but you do need to submit it by this date. And so what you would do is you would click on this. And so you would go to submit assignment and you would upload your document. So again, if you have this in one file, you can upload it as one single file that has both ID sheets, or if you need to upload two, that's fine, but you need to upload both the nutrient agar and the McConkey agar. And so you will upload this, and then I will grade it, and I will give you the next part of your project. And so you need to make sure that you submit it before that due date. That is the final date in which you can submit it. So you need to submit it by that day, and then I can give you the next part of your assignment. So after you submit your assignment, notice that the assignment is graded. So you will have a grade here. So when you're on the submission, you'll be able to see the grade. And then notice that I gave you the next part of this test. So the next part would be to do a gram stain. And so I gave you two images. One would be your gram positive and one would be your gram negative. And so this is where you would get that image to do the gram stain. So after you finish the describing your isolation, what you would normally do is after you do your isolation, right, after you separate your two types, then you can gram stain. I can't gram stain from the broth because the broth is going to have a mixture of both my gram positive and my gram negative. So I have to streak for isolation first. So you're going to analyze your streak plate first. Then is going to come the gram stain. And so the gram stain has been performed. So I told you what's been done. A gram stain has been performed from your nutrient agar plate only. So again, you want to go back and kind of look at McConkey and decide, why didn't I gram stain from McConkey? There's a reason that we can't use the McConkey plate. We had to use the nutrient agar plate. So gram stain has been performed from my nutrient agar plate. Each of the two types of colonies, so I have to identify the two types, right? Because remember that in my broth, I had a mixture of gram positive and gram negative. So I'm going to have two distinct colony types. So each of the two types of colonies were picked, and a gram stain was performed for them on the same slide, but in two separate smears, so not placed in the same puddle of water. You might recall when we did our gram stain, we had one puddle of water, and we mixed both our gram positive and our gram negative in the same puddle of water. But for this project, we wouldn't want to do that. We don't want them in the same puddle. We want them in two puddles of water that are side by side, because one of them we would call colony A, whatever that colony looked like. We would want to see, is that the gram positive colony or the gram negative? And so side by side, we should have the other colony. Now, you need to think about why we do both drops of water, both bacteria emulsions, on the same slide. There's a question for your report that has you address that. And so you want to think about what is so important, what's the important step in the gram stain, and why would we want to do it on the same slide versus doing it on two separate slides? What could be a potential problem if you did your gram stain on two separate slides? So there's a reason that we would do this, two puddles of water, side by side on the same slide. So I'm telling you that's what we did. And so it says, for both organisms, you must note the gram reaction, so whether it's gram positive or negative, what the morphology is like, so what is, again, the shape, the arrangement, is it in clusters, is it in chains, and any identifying characteristics that are observed under the microscope. This is as far as you'll go for your gram positive. So when you get to this step, you will propose a genus for your gram positive. That's it. That is all you have to do for the gram positive. The gram negative is going to continue on. We want to identify the gram negative down to the species. So for your gram-negative, the third part of this project is identification of your gram-negative unknown, and you will identify your gram-negative using biochemical tests. So all of those biochemical tests that you've been learning, those will be what are used to help you identify your gram-negative. And so the first thing that you would need to do would be to do an IMVIC. And so you will get results for an IMVIC. And so just like where I gave you the gram stain pictures, also included on your streak plate submission, you will also have an IMVIC result. And so you will use those pictures and you will read out those tests and then do the next test on the list. So the idea behind this would be that if you were actually in person, you would, on the first day we start this project, we would give you a broth, you would streak for isolation. So you would streak on your nutrient agar and your McConkey agar. You would take those isolated plates and your gram stain would be from the nutrient agar plate. Because again, the nutrient agar is going to have both types. However, you want to think about what plate should you use to identify the gram-negative. Do I want to use my nutrient agar plate, which has both bacteria, or do I want to use my McConkey agar plate? And so what you would do in a normal class would be after you got your streak plate, then you would perform your gram stain and you would set up your IMVIC. And so when you submit your ID sheets for your streak plates, that would be when I give you the results for your gram stains, and I'll give you the results for the IMVIC. You would need to read out your IMVIC, and then in theoretical terms, you would submit your results. So let me show you how you would read out your IMVIC. So when you go to do your readout for your IMVIC, right, I'm going to give you your picture and you're going to look at that picture and then you need to figure out what the next test would be. So the way that you do that is if you go to these unknown identification flowcharts, you would click on this and it's going to bring up a document that looks like this. And so notice that depending on what your IMVIC results are, that will tell you your next test. So for example, let's say that I got negative, positive, negative, positive. So let's say it was indole negative, MR positive, VP negative, and citrate positive. If this is my readout, then I would go to figure 3. So different people are going to be on different flowcharts. So if I'm figure 3, I'm going to scroll down. And here is figure 3. So again, notice my IMVIC, negative, positive, negative, positive. So these are the possible organisms that would fall in that category. So now we use what's called a dichotomous key. And so what would happen is that when you do your IMVIC, so when you get your picture of your IMVIC results, you are going to fill out a biochemical sheet. And you're going to have one for indole, you're going to have one for your methyl red, you're going to have one for VP, and one for citrate. And so you will upload all of your IMVIC biochemical ID sheets and your gram stain to the submission. And I'll show you that in a minute. When you submit that assignment, in the comments section, you need to tell me what figure you're on. So which flowchart are you on based on your IMVIC? And then you need to tell me what is the next test that you need the results for. Because you need to figure out what your next test would be. This would be like if you were in person. If you were actually doing your IMVIC, you would then, based on your results, then you would set up your next test. So if I did my readout and this is my result, notice my next test would be an ornithine decarboxylase test. So if I were doing this in person, I would then inoculate an ornithine decarboxylase tube, put it in the incubator, and then 48 hours later, look at the result, and then set up the next test. So when you guys do this, when you get your INVIC results and when you submit your ID sheet, you need to tell me what figure you're on, and you also need to tell me which result you need next. So you would say, Dr. Wells, can you please give me the result for the ornithine decarboxylase test? And in that comments, after I check your ID sheets, then I would give you the next result. Now, if my result is positive, notice ornithine decarboxylase, positive is purple, right? If my result was my positive, then the next test would be a TSI. And then if the TSI was positive, the next test would be a phenylalanine deaminase. And so I would do all of these tests until I end up with my organism. So let's say it was positive for phenylalanine deaminase. Well, then my organism would be Proteus mirabilis. And so that would be my identification of my gram negative. So let me go back and show you, once you get your pictures for your gram stain and your INVIC, and you write up your sheets for that, where do you submit them? So notice that there is a part for you to submit your INVIC and your gram stain ID sheets. And so this would be where you would submit that. And so once you submit your ID sheets for the gram stain and for your INVIC, once those are graded, then I will give you, and you ask me for the next result. So again, you would say, Dr. Wells, could you send me the result for ornithine decarboxylase? So in the comments on this, then I would give you that result. Then you would look at that picture, and then you would do the readout. You would then submit that information for biochemical test ID sheet number 1. So this biochemical sheet would be for the ornithine decarboxylase. And so you would have an ID sheet for that. And when you submit that, then you would ask me, based on that result, Dr. Wells, can you send me the result for TSI? And so I would give you the result for TSI here. You would then submit an ID sheet for your TSI. Then when you submit ID sheet number 2, again, you're going to tell me what test do you need next, which result do you need next. You would ask for that. I would respond. Then you would write the ID sheet for the last one. And so this is the last biochemical ID sheet. Now notice, basically for each week, one of these sets of ID sheets is due for each week, and they are due Friday at 11:59. So that's consistent. Every week, starting October 23rd, every week you have a submission at Friday at 11:59 p.m. And you want to do that. The faster you get those submitted, the faster you're going to get the next test result, so that you have longer to work on those ID sheets. If you wait until the last minute, well, you're not going to have as much time to do the next part of your test, because you have to wait for me to grade the part that you submit in order for you to get the result for the next part. So again, try and submit these as soon as possible so that I can grade them, give them back, and that you are ready for the next part of the test. The busiest, probably the one that's going to take you the most time, is this INVIC gram stain ID sheet, because there are 4 ID sheets for the INVIC and then another one for the gram stain. So one other thing to be aware is that for some of the flowcharts, there might be a choice of 2 tests that you could choose between. For example, it says on the bottom right side of figure 2, there are 2 choices. You could perform a phenyl red glucose with gas or an ONPG. You could pick one of those to perform. And so this is your project, so make sure that you keep on top of your timelines, the response times, and the deadlines. You need to pace yourself and make sure that you submit things early so that you have enough time to get a response so that you can submit the next part of your test. And so you would do your biochemical test until you identified your gram negative. So in terms of your identification sheets, you're going to have identification sheets, and you're going to have, if you look down here, the ones that you're going to need, you need one for the nutrient agar streak plate, one for the McConkey agar streak plate, one for the gram stain, and then you're going to need ID sheets for your gram negative unknown. So basically, starting with your IMVIC, a separate test sheet for indole, a separate one for MR, separate for VP, and a separate for citrate. And then your tests, you're going to need a biochemical sheet for each of the tests that you do after that for your gram negative. Now, in terms of those identification sheets, do not use first person for this section. You don't want to say, I would inoculate a tryptone broth using a loop from the McConkey. You don't say I. You would simply say inoculate a tryptone broth using a loop from the McConkey plate. So you want to include what plate you're using to do that test, what tool you're using to do your inoculation. And again, you don't have to say every little detail in terms of aseptic technique. Assume the person who is doing this knows aseptic technique. You want to describe, though, how do you do your tests. So don't use first person. Make sure that if you use lists, like if you use bulleted lists, that those bulleted lists are still in full sentences. Refrain from using contractions, can't, won't, etc. And again, don't use first or third person, I, we, me, they, us. For each of your identification sheets, you should record the following information on the ID sheet. And so you would include, again, the name of the test, the purpose of performing that test and why it's important, the procedure that is followed, and any appropriate controls, if there are any, in your own words. Make sure that you talk about both the setup part of the test, so how you inoculate the tube, and then if there's anything that you have to do for the readout. So like, for example, if I was looking at the phenylalanine deaminase. So after 48 hours, take your tube out and add 5 drops of ferric chloride to the slant, rock back and forth, and look for a color change. So don't forget that there is a part sometimes after the incubation, that there are steps that you have to do after you take your tube out. So if there are steps, don't forget to add that in. My other hint would be for the MRVP, remember that you inoculate one broth and then you separate it into 2 tubes. So don't forget that step when you separate it into 2 tubes. And then what do you add to the tube for the methyl red? What do you add to the tube for the VP? So those are my hints in terms of the procedures. So make sure, again, in your procedures that you include what plate are you picking up bacteria from? Which plate are you using to inoculate? What utensil are you using? So are you using a loop or a needle? How long do you inoculate, or how long do you incubate that tube? And what is the temperature for the incubation? And then don't forget to add any reagents if there are reagents. You need to include your test results, including the appearance, so color drawings or digital photos, and any unexpected observations. The ones that you definitely need to use the actual photos, you need to use the actual photos for sure for your gram stain and for your streak plates. I need to see the actual pictures so that I can analyze the way you've described it. Lastly, your interpretation of your test results, including a full description of what products, if any, were detected, and how and why those products contributed to the final appearance of the test. Also indicate any reagents or pH indicators that are used for in this test, and why the test is negative if negative. So if your test is negative, you should include what products you were looking for, even though you didn't get them. What products were you looking for, and why the test is negative. So again, this is like your biochemical sheet. However, do not just put substrate equals, enzyme equals. Describe it in complete sentences. So this section, those identification sheets do not need to be typed as long as they are legible. So as long as I can read it, it could be handwritten, meaning that if you wanted to print these blank sheets and then handwrite on them and upload them, I am fine with that as long as I can read it, or they can be typed. Again, it needs to be in complete sentences. Don't put things in bullets without complete sentences. So make sure that your identification sheet is in complete sentences. And so each identification sheet is worth 3 points for a total of 30 points. Now, one of the things that I want to point out is that on the syllabus, it says that your unknown project is worth 30 points, and then 20 points are professionalism and participation. I have updated that. It's 40 points for the unknown project and only 10 points for professionalism and participation because we aren't in lab. And so the professionalism points, meaning if you follow the rules in the lab, don't really apply here because you're not actually in the lab. So I have decided to make participation and professionalism worth only 10 points. And your report, your unknown report, is worth 40 points. But 30 points are those identification sheets. So make sure you're very thorough on your identification sheets so that you get full credit. The last part of the report is going to be the actual report. And this section should be in full, complete sentences. Bullet points or lists are not acceptable for this portion of the paper. Refrain from using contractions, can't, won't, etc. Don't use first or third person. Again, everything should be inoculate a tryptone broth with a loop, etc. So no first or third person. So the first part of your report is where you're going to describe your isolation. And so your isolation description is worth 2 points. And so it says, begin by describing what was involved in separating both unknown bacterial types on nutrient and McConkey agars. So basically, what technique did you do to separate bacteria? Why were both types of media used? So what was the purpose of using the nutrient agar versus what was the purpose of the McConkey agar? What number were you given? So you're going to record what number you were given. You want to describe, were your colonies well separated on the nutrient agar plate? And then you're going to describe your colonies. So both your colonies for the nutrient agar, and again, use your lab manual. And then you should also describe what your colonies look like for the McConkey agar. So describe the colonies on both plates. Remember that your nutrient agar should have 2 types of colonies that you're going to describe. There are 2 separate types of colonies. Make sure that you describe both for the nutrient agar and also describe the colony on the McConkey agar. The next part of your report is going to be your description of your gram stain. So this is 4 points. So describe the appearance of the 2 types of bacteria that you saw in your gram stain. Be sure to include morphology, shape, arrangement, and whether endospores were present. So for example, if my gram positive, I don't want to give you the color because I don't want to give you which one is gram positive and which one's gram negative, but let's say I saw spheres and they were in chains, right? So if they're spheres and they're in chains, I could propose that the genus would be Streptococcus, because Streptococcus is a genus. It is gram positive, and that's what it would look like. in terms of endospores being present, you want to think about what would endospores look like in a gram stain. Meaning, in a gram stain, would you expect the endospore to be able to take up the dye? Why or why not? Think about the way we do our gram stain. So you want to think about what would an endospore look like in a gram stain. Would it be colored? Would it be colorless? And so when you're describing your gram stain, be aware that you might have an endospore producing bacteria. And if you recall, there are two main genus that you could propose if you had an endospore producing bacteria. And my hint is to think that only one of those two genus could be applied in this case. And so I want you to think about that. If you see possible endospores on your gram stain, remember that you are incubating those plates in the incubator. So that should rule out one of those two genus. And that leaves you with one possible genus that you could propose. So keep that in mind and think critically if you have an endospore producing bacteria so that you propose the correct genus. You also want to include why was it important to perform the gram stain of the two organisms on the same slide. So why did we do them side by side on the same slide? And again, think about what is the most important step during that procedure and why is it beneficial to do it on the same slide versus doing it on two separate slides. So you're going to describe that for your gram stain. And then lastly, you're going to describe your results for your gram-negative unknown. This is also worth 4 points. And so it says, summarize your tests and observations briefly following the flowchart formats illustrations posted on Canvas. Based on your own data and the dichotomous key, determine the identity, genus, and species of your gram-negative unknown. So for this part, when you're doing this, you want to be very brief in your summary. You don't need to go through your biochemical sheet for every single test. And what I mean by that is when you're summarizing your results, you could be very brief and say, when performing an INVIC, the results that were seen were that the organism was indole negative, MR positive, VP negative, citrate positive. According to flowchart number 3, whatever flowchart you're on, the next test that would be set up would be the ornithine decarboxylase. The ornithine decarboxylase result was positive, which then led to the TSI test. The TSI test was yellow with black, which is positive for sulfur reduction, and that led to this test, so on and so forth. So it's very brief. It's basically just summarizing your results and then kind of your flow of the experiments that you did that led you to your identification of your bacteria. You're going to get to your species of your gram-negative. Then you're going to conclude your report with a paragraph of a short description of your identified bacterium. So where the bacteria is normally found, whether it's medically significant. If the bacterium is medically significant, indicate the diseases or conditions that are caused by the bacteria. This section, you're going to have to do a little bit of research. So make sure you cite your information in this section in MLA or APA format. You must have references for the information that you're providing. Do not use Wikipedia as a source. That is not a reliable source. So make sure that you find reputable sources to describe those bacteria, where they're found, and the diseases that they cause. Your report should be typewritten. It should be in 12-point font, double spaced. Attached to your report, include a drawing or a photocopy of the flowchart that you used and highlight the steps that you followed. And so what that means is that you can take your flowchart and simply just highlight the steps that you took that got you to your bacteria. You could print this, highlight with a regular highlighter, and then scan it again. You could do it on the computer, however you want to do it, but I want you to include your flowchart that you used and then show me the steps that you took that got you to your bacteria. And so that's what that means by the flowchart. The report is worth a total of 40 points, and it's due on Monday, December 7th by uploading into your Canvas assignment by 11:59 p.m. And so this is just an overview of basically the tests that you would do if you were doing this in person. So day 1, you would streak onto Nutrient Auger and McConkey Auger. The next class period, you would come in, you would get your plates, and you would gram stain from your Nutrient Auger, as well as you would set up your IMVIC. We would typically also have you streak onto a slant just because it allows the bacteria to be kept longer, but obviously for the purpose of this, we didn't need to do that. The next class period, you would read out your IMVIC, and then you would set up your first biochemical test. So using your flowchart, you would set up your next biochemical test. And then you would continue your biochemical tests until your gram-negative bacteria is identified. And then your report would be due on Canvas on December 7th at 11:59. And so again, this would be, notice your unknown project submissions. This is where you're going to submit each of these parts of the test. So you're going to submit your Nutrient Auger and your McConkey Auger streak plate ID sheets here. So you're going to have 2 for this one. You're going to submit your IMVIC and your gram stain ID sheets here. This is the biggest day in terms of the number of points as well as the number of ID sheets, because you're going to need 5 total. 4 for your IMVIC and 1 for your gram stain. So in your gram stain ID sheet, the other thing you should put in your interpretation is, why do gram positive and gram negative stain differently? So why is gram positive the color that it is, and why is gram negative the color that it is? So I would include that in your interpretation. So you're going to submit that one by the due date. And then don't forget, for each of these tests, when you submit this, you need to tell me which test result you need next. And then in the comments is where I'm going to provide you with that result so that then you can do the next day. And so the more you keep on top of this, the better, because that means you'll get your results back quicker and you'll be ready to do the next part. And then the last part of this is your actual report. Your typed report is going to be due on December 7th. You don't need to resubmit your ID sheets here. You've already submitted the ID sheets here. This is just that typed part of the report. The ID sheets were already turned in. So this is just that typed report part. And so this would be where you would submit this. So all of your unknown project information is on the top, and then these are your submissions. So again, your info is where you're going to find your number. So look at the list and find your name and figure out what number you have. You will also be able to find on that page your picture for both of your streak plates. This is the directions so that I've been going through in this video. This is your blank identification sheets. This is an example of an identification sheet so that you could see what that would look like. And then these are your flow charts. And so this is how we would go about doing our unknown project. And so this is going to be a remote or a simulated version of your unknown project. Unfortunately, you don't get to do this hands-on, but we will run through the test just like we would if you were in person. And so this is how we do our unknown project.